Hi everyone, welcome to your Chapter 18 video. Today we're going to be covering Chapter 18, which goes over control of gene expression in bacteria. But before we jump into that, if you're watching this in the fall of 2020, I wanted to give you a quick reminder that this Friday, November 6th, you will have your last regular exam of the semester, okay? This exam is going to cover the following chapters. Let's go through what we will be talking about on the exam. So it's going to cover chapters 16 through 19, which seems like a lot, but let's break it down topically. 15 through 19. Chapter 15 was all about DNA synthesis, okay? So that will be on your exam. Chapters 16 and 17 kind of work together to talk about the processes of transcription and translation. Chapters 18 and 19 are going to both cover this topic of gene expression, which I will define for you in a moment. But essentially, Chapter 18 is going to look at gene expression in prokaryotes, and 19 is going to look at it in eukaryotes. And each of these will be one lecture apiece. So essentially, you've got three main areas of concept to focus on for your exam, okay? So be prepared Friday, November 6th to take your last regular exam for me. After that, you will have one more homework. You will have one more quiz. And then you will have the final. And remember, the final is worth 200 points, so it's essentially worth the same as taking 2 exams. Okay? All right, moving on to Chapter 18. So Chapter 18 is going to focus on controlling gene expression, and specifically there are 3 things that I want us to discuss in this chapter, okay? First, we're going to define this term, gene expression, okay? Second, we are going to talk just a second on why we want to control gene expression, because this is the topic of this chapter, controlling gene expression. So why control it? Why is that important? But the majority of this chapter is going to be spent showing you examples of how gene expression is controlled in bacteria. And these are classic examples that helped us to understand a little bit about the process of controlling gene expression, period. Okay? So let's chat for just a second about what gene expression is. So gene expression really can be thought of as let's put it this way, when and what level gene products are made. Okay, so when and to what level are gene products made? Now, what do I mean by that? Let's go back to our central dogma for just a second. So you'll recall that our central dogma states that DNA is copied via transcription into RNA. RNA, through translation, is converted into proteins. And these proteins do the work of the cell so that we end up with traits, right? So gene expression is simply a word that we use to describe how much protein we make of a particular gene. Remember that genes encode proteins. How much of this protein do we make? And is this protein active or is it inactive? Is it actually doing the work that results in traits? So that's what gene expression is, okay? Now, why do we control gene expression? Because remember, if you think about a bacteria, let's draw ourselves a little bacteria. Here's our bacteria. And you know, it's got like one circular chromosome. That's what I'm denoting here. So that's our DNA. If we were talking about a eukaryotic cell, you would have in the nucleus a whole bunch of DNA, and this DNA would be in the form of multiple different chromosomes, right? Each cell, let's put DNA here as well. The DNA in each cell, you can think of that as a complete instruction book. So let's just put this as complete set of instructions. So the DNA in these cells represents a complete instruction book on how to make a whole nother cell, or a whole nother organism if you're talking about a multicellular eukaryote, right? But you might not need to activate all of those genes all of the time. Here's what I mean by that. Let's just envision a cell that we've plucked off of your eyeball. The nucleus of that eyeball cell has DNA that basically is the entire set of instructions on how to make an entire another version of you. But your eyeball cell doesn't need to use all of those genes. It only needs to turn on the subset of genes that are relevant to the functioning of the eyeball, right? It doesn't need genes that are relevant to the liver. It doesn't need genes that are relevant to the stomach. If your eyeball suddenly turned on genes that were relevant to your stomach, you might start making stomach acid in your eye, and that would not be any fun. So while you have all of the genes in each and every cell that tell everything about how to make a new version of you, each of your cells is only accessing a small percentage of those. So if you think of your DNA as a giant instruction book, cells are only looking at the chapters that they need in order to function properly. Now, that's also going to be true in bacteria. While bacteria are not multicellular, they don't need every single gene that is encoded, that encodes a protein here all the time, right? So, for example, if you have a bacterium that is growing up in a solution full of glucose, then it only turns on the genes that you need to digest glucose. But if you move that bacteria to another growth medium, maybe this growth medium has lactose in it and there's no glucose, then it's going to turn on the genes that it needs to digest lactose, right? So why not just have all genes on all the time? Well, really, it's because it wastes energy, right? It would waste energy if we expressed, if we'll put, if all genes expressed, if we made proteins out of all the genes all the time. Or another way to think of that is to say, if all the genes were turned on, that would waste energy. And that would be a problem if you're a bacteria, right? You're trying to conserve your energy because you're trying to occupy and populate maybe a region of the gut or something. All right. Now, remember that the whole point of this entire process is to end with an active protein that ultimately does work in the cell, and this work results in the traits that we see. So the goal of turning on any gene is ultimately to have an active protein. But we can regulate whether a gene is turned into an active protein at 3 different places, okay? 1, 2, 3. The first place that we can regulate whether or not a gene is turned into an active protein is at the level of transcription. So this would be called transcriptional control. And this is going to consist of mechanisms that maybe turn off RNA polymerase or block RNA polymerase from copying DNA into RNA in the first place. Okay, might also involve blocking promoters of genes or something like that. All right. The second place that we can control whether this gene is turned into an active protein. I'm going to do this as the active protein because if the protein is active, you have a trait. So the second place that we can exert some control is in translation, right? Once we've made our mRNA, I'll put translational, control. Once we've gone through and we've made our mRNA, the cell can choose whether or not to turn that into a protein. And there's several different mechanisms that you could use to exert translational control. You might digest your mRNA, or you might somehow alter like the availability of the mRNA to ribosomes. If you've already got the mRNA, you can do things that prevent it from being turned into protein. Now, what if you've made it all the way through from DNA, you copied it into mRNA, and then you translated your mRNA into protein? Well, you might be able to also exert some control over whether this protein is active or inactive using what we call post-translational control mechanisms, okay? You guys might remember some things we talked about, like you can add chemical groups to turn proteins on, or you can add chemical groups to turn proteins off, and that would determine whether the protein is active or inactive. So those are examples of post-translational control mechanisms. So let's look at it here a little differently to kind of drive home this idea that there are 3 levels where we can control gene expression. And gene expression, we can really think of it as turning on genes, or we can think of it as turning genes into active proteins, right? That's really what gene expression is. All right, so as I mentioned, the first thing that we can do is we can use different mechanisms to determine whether the DNA is transcribed into RNA. So we might do things like have mechanisms that block promoters. We might block the action of RNA polymerase, something like that. But these are all going to be mechanisms that prevent copying of the DNA into RNA in the first place, okay? Now, the nice thing about exerting control at this level is it saves the cell a lot of energy, right? If you don't start this whole process, if you don't start going from here to here in the first place, then you're saving the cell some energy, aren't you? Okay. The next place that we can exert some control is at the translational level. So you can do things like you can change the lifespan of the mRNA, okay? So you can do things like mRNA degradation, we'll put degrade, okay? You can maybe change the availability of the mRNA to the ribosomes, but once you've gone through this whole process of creating an mRNA, you can initiate different mechanisms that change whether or not the mRNA is turned into protein. And then the last thing that you can do is you can go all the way through copying the DNA into RNA. You can then translate that RNA into protein, but post-translational control includes mechanisms that activate or inactivate proteins. So we've seen examples of this before. So here's an example of a protein that we've gone from DNA to RNA. We've taken our mRNA and we've translated it, and now we have this protein right here. But in this example, this protein is inactive. You may remember that I told you that a very common mechanism to make proteins active, or to turn proteins on, if you will, is to add chemical groups. When you bind something to a protein, you usually change its conformation. And a common chemical group that gets added to proteins to turn them on is a phosphate group. And if you remember, this process was called phosphorylation. So you can phosphorylate proteins to turn them on or off. Now, I mentioned that if you shut the process down before you ever get started, that saves the cell some energy. But so you may be asking yourself, like, why would you ever get down to this point and go to all this trouble of creating an inactive protein? Well, if controlling things on this end saves energy, controlling things on this end saves time, right? Let's think about this. Let's think of an example. So we're going to talk in a minute about bacteria using different sugars as energy sources. They can use glucose. They can also use lactose. I'm going to put lactose over here. So let's pretend that we have a little E. coli here, and we have it submerged in a growth medium that contains glucose. That's wonderful. But what if we move it into a new beaker, and this new beaker is going to contain lactose? No glucose is available. Well, if we are controlling things on this end, in order for that little bacterium to be able to use this lactose, we have to dial up this entire process, starting with copying the DNA into RNA, then translating it into protein, and then activating any proteins that might need activating. So while we've saved ourselves some energy, there's going to be a lag time involved before we can actually get around to digesting this lactose. On the other hand, if you've gone through this entire process and you just have these inactive proteins sitting around, then if you move this E. coli into a new environment, all we have to do is flip this switch, turn this protein on, and we very quickly are able to use our lactose. So there's advantages and disadvantages to being on either end of the spectrum. And of course, this area here represents an intermediate for both, right? It's intermediate in the amount of energy that it uses, and it's intermediate in the amount of time it would take for the cell to respond to a new environment. Now, to illustrate these ideas of controlling gene expression, we're going to be looking at the metabolism of lactose in E. coli, okay? When I say metabolism, what I mean is breaking down lactose to turn it into ATP for energy, okay? So that's the model that we're going to be using. And we're mostly going to be focusing on transcriptional level control, controlling when these genes that metabolize lactose are transcribed, okay? So, but while we're going to focus on this end of things, we'll also talk a little bit about this end of things, but please remember that there are examples of translational control that also exist, okay? And we might talk about those in chapter 19, and we talk about those a lot in genetics class as well. All right, so in order for an E. coli to digest lactose, it's got to do a couple things. So I'm going to just draw an E. coli because that's kind of going to help me think. Normally, what an E. coli loves to eat is glucose. Okay, its normal mode of operation is to use glucose as its primary food source. And this makes a lot of sense if you think about it, because if you'll remember back from when we studied cellular respiration, glucose is the substrate for cellular respiration. Now, bacteria don't go through cellular respiration because they don't have mitochondria, but they do go through the first part of that process, which, if you remember, that was called glycolysis. So these bacteria do undergo that set of chemical reactions that break down glucose and get just a little bit of ATP. So the reason that E. coli are going to have a preference for glucose is because it plugs right in to this glycolysis process. Now, while E. coli prefer glucose, they can also use lactose. And y'all probably know that lactose is found in dairy foods. And lactose, I'm going to just write the word lactose here. Lactose is a disaccharide. And that is a vocabulary term that you might remember from way back talking about carbohydrates. So if you remember, carbohydrates can be broken down into monosaccharides A disaccharide is made up of two monosaccharides. If you break lactose into its two constituent parts, you would find that it's made up of a molecule of glucose, as well as another molecule that's a closely related sugar called galactose. It looks very similar to glucose. It's got some OH groups that are in a little bit different place, okay? So they're both 6-carbon sugars. So in order for the bacteria to eat this lactose, it's got to break it down. And so in thinking about breaking down lactose, there's really two pieces that we need. First of all, we have to get lactose into the cell, okay? And in order to do that, we're going to use a protein transporter called galactoside permease, or you are welcome to just call this thing permease for short. Okay, so permease, what it does is it functions like a tunnel that gets inserted into the bacterial cell membrane, and it allows lactose into the cell. Now, the important thing to remember is that this is a protein. That means that it is encoded in some DNA. There is a gene that codes for this permease protein, okay? We're going to talk about that gene in just a second. The second thing we need, once we've inserted our little tunnel to get lactose in, is once we get lactose into the cell, remember I said that lactose is a disaccharide. And so we're going to have to use some kind of an enzyme to split, so we'll make this an enzyme, to split this guy into two individual sugars, glucose, I'll just put GLU for glucose, and galactose, and I'll put GAL for galactose. So that's what you're seeing here. Here's our lactose. It's got a glucose and a galactose. It's going to go through our transporter protein, permease, and then an enzyme called beta-galactosidase. Let me just write that down for you here. Beta-galactosidase is going to be responsible for breaking this apart into two individual sugars. And it's these guys that the bacteria can use to make ATP, okay? So we have a protein here in permease, that's protein number 1 that we need in order to be able to break down lactose, and protein number 2 that we need is beta-galactosidase. Each of these are proteins, which means that there's a gene that codes for each of them, all right? So let's put a pin in that. We're going to come back to that thought later. Now, what researchers very early realized is that if you were dealing with bacteria, if you were dealing with E. coli, and you fed them only glucose, and you looked at what enzymes were present, if there's only glucose, there is no permease in a bacterial cell, and there's no, I'm going to put beta-gal for beta-galactosidase, okay? So those are not made. If you're a bacteria, you do not make these two guys, because let's go back here. You don't need them, right? All this whole thing that I talked about was to help you eat lactose if you're a little bacteria. So in the presence of glucose, it turns out that there is no permease and no beta-galactosidase made. If you have lactose, then yes, you have both permease and you have also got beta-galactosidase. What's interesting is that when researchers grew bacteria in a medium that contained both glucose and lactose, then they found that, let me draw that out here, that situation ended up in no permease and no, I'm going to put beta-gal. Interesting. So even though lactose is present, bacteria are preferentially eating up the glucose, and they're not even making the enzymes to make lactose, okay? So they are controlling their gene expression. They're only making these two proteins if no glucose is available. Okay, how can that happen? And that's what we're going to walk through for the remainder of this lecture. Now I'm thinking about how that can happen. It turns out that in bacteria, metabolism of lactose is transcriptionally controlled. In other words, whether the DNA is copied into RNA is where we're going to control whether or not we make those two proteins, okay? And there's two different types of transcriptional control that we're going to cover today. There's negative transcriptional control, and there's positive transcriptional control. And it's very easy. Negative transcriptional control turns off transcription. Positive transcriptional control turns on transcription. So we're going to learn in this lecture how bacteria turn off transcription of these two genes, the one that codes for permease and the one that codes for beta-galactosidase. And then we're going to learn a little bit about how the transcription is turned on, or even let's just say turned up, okay? Now, I've oversimplified things, and you're going to see that these lines get a little bit blurred, but I think you'll see what I mean in just a moment. So to get this party started, let's draw ourselves a stretch of bacterial DNA. And I'm going to make this nice and long, okay? So if you're taking notes with me, give yourself some space in your notes to make a nice long stretch of DNA. This DNA block that I'm drawing, understand that it is meant to represent double-stranded bacterial DNA, okay? And theoretically, this would continue on in a big old circle, but we're just drawing one section of it. And in this section, let's put in some genes, okay? So genes start with a promoter, so I'm gonna put a promoter here, and I'm gonna designate that with the letter P for promoter. I'm also gonna put another promoter up here, okay, for another set of genes. This gene right here, we're going to label as LAC-I, and I'll explain what that is in just a second. This promoter is actually going to be a single promoter for an entire set of genes. So we're going to have one promoter that actually functions as a binding site. Remember that both promoters function as binding sites. Just in case you need a little refresher, this is where the RNA polymerase is going to bind to transcribe these genes. So you can get RNA polymerase binding here, or you can get RNA polymerase binding here. If RNA polymerase binds here, it's actually able to transcribe 3 genes, but I want you to draw 4 boxes, okay? It can transcribe a gene called LAC-Z. It can transcribe one called LAC-Y, and it can transcribe another one called LAC-A. And I bet you can guess, based on this terminology of lac, that these are all genes that are going to be involved in metabolizing of lactose. That's where that lac comes from. As a matter of fact, this gene right here, let's see here, let me make sure I'm telling you correctly about which gene is which. This gene, I will tell you which codes for which, hold on. Yes. So LAC-Y, hold on, I just want to make sure I get this right. I always get these backwards. Yes, okay, I am right. Okay, so LAC-Y codes for permease. Remember, permease was that protein that lets lactose into the cell. LAC-Z codes for beta-galactosidase. I had to break that up. LAC-A codes for a third gene related to lactose metabolism that we're not really going to talk about, okay? Now, the only region that I haven't really filled in, this is just going to be some regular old DNA here, so leave this blank. But this region here has a special name. It's called the operator. Okay. And it's going to have a special function that I'm going to talk about in just a second. All right. This entire unit, notice that all these genes have lac, 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 lac. These are all related to lactose metabolism. And in bacteria, a group of genes that have a related function and that are frequently transcribed. Hi, everyone. So what I was saying before Zoom cut me off earlier today is that if we look at this entire segment of DNA here, okay, these are all genes that are related to lactose metabolism. And so we actually think about these genes as a unit, and there's a name for this unit of genes that are all related in function. Those are called operons. So we have this gene up here, this gene here. This is all part of the operon. Another feature that I'd like to point out is that remember that promoters, like I have for you here and have for you here, remember that, as I said earlier, these are binding sites for RNA polymerase. So we know that RNA polymerase can bind here at this promoter and then transcribe this LAC-I gene. Here, RNA polymerase could bind to this promoter, and if it transcribes all the way through, notice we only have one promoter, but 3 genes that are related in function. This beta-galactosidase, permease, and this LAC-A gene, which is involved in transporting some of the waste products of lactose metabolism out of the cell. So when we have a situation where one promoter causes transcription of many genes, the term for this is that these are polycistronic genes. Okay, so what we have here is a situation where we have a gene that codes for a protein, a gene here that codes for a protein, and a gene here that's going to code for another protein that we're not going to talk about right now, okay? So hopefully you see that we have one gene, one protein correspondence. So what I'd like to do is walk you through what happens under a normal situation when glucose is present in the bacteria's environment and it's available for the bacteria to use as a food/energy source. So let's start at this end of the DNA. What normally would happen is that RNA polymerase, and we'll just draw it, RNA polymerase, I'm just going to kind of draw it like this, is going to bind to that promoter. And remember, in order for it to bind, it actually is going to be RNA polymerase plus the sigma factor. Remember that sigma factor binds to specific sequences in the promoter, and that in turn allows RNA polymerase to bind. And when that happens, RNA polymerase is going to move through and transcribe this gene. This gene codes for a protein called the repressor. All right, so this repressor is going to be transcribed and it's going to be translated. And when it's all said and done, it's going to look something like this. Okay, and I'll just color this guy in for us. Now, this repressor protein is transcribed all the time, okay? It is transcribed and it's translated all the time. So this gene is always turned on. When we have a gene that is always turned on and it's really not, its gene expression is not controlled. It's just always on, always being transcribed, always being translated, and we end up with active protein all the time. We say that this particular gene, let me change colors here so y'all can see, is constitutively expressed. So we're going to write C-O-N, constitutively expressed. So this repressor here is constitutively expressed. It's always turned on. We're always going to have this protein available. Now, what this protein is going to do is it comes along and it recognizes sequences in this area here called the operator. So this repressor is able to latch on to the DNA. You can hopefully kind of see why I drew it the way I did. So we're going to have a kind of clamping situation here, where this is kind of straddling this region of the DNA. So this repressor, I'm going to put repressor, binds to the area of the DNA called the operator. Now here's why that is significant. Notice where the operator is. The operator is right here, kind of in front of, just making this a little more colored in for you guys, kind of in front of this promoter. So here's what's going to happen. If RNA polymerase, let's change colors here. If RNA polymerase of sigma binds right here, which then allows RNA polymerase to bind right here, RNA polymerase is effectively blocked from moving forward because, as I like to say, there's a car parked in the middle of the road. This protein here that's bound to the operator is literally blocking the progress of RNA polymerase to move across this DNA and transcribe this suite of genes, okay? So when glucose is present in the cell, what happens is this repressor gets transcribed and translated. Here's our repressor. It comes along and it binds to this region here, the operator, okay? Now let's look at it in a little bit better rendering. So lactose is absent in the cell, which we're gonna say in this case, not only is lactose absent, but we're gonna say that glucose is present. What happens is this LAC-I gene gets transcribed to make this repressor protein. This repressor protein then binds to this region of the DNA called the operator. And you can see here, it says repressor bound to operator, it's blocking the movement of RNA polymerase to go through and finish transcribing this region here. Now, let's go back to our original slide that I did here. Now, let's switch scenarios a little bit, and let's say that the bacteria has used up all the glucose. Now we're going to feed it lactose. So now lactose is going to be present in the cell, but glucose is not. Well, the interesting thing about lactose is lactose can bind to the repressor. Now, I want you to think about what happens to proteins when something binds to them. When something binds to proteins, it almost always changes their shape. So when the lactose binds to this repressor, it changes the shape of this repressor and it causes the repressor to pop off. I'm going to put that in quotes because that's not a very scientific term, but you know what I mean. So it causes the repressor to pop off. If we remove this repressor, then you can see that RNA polymerase will be able to transcribe all the way through here. So what has effectively happened is that lactose being present in the cell allows transcription of the genes that are involved in lactose metabolism. Now you may be asking yourself, wait a minute, let's back up here. For lactose to get into the cell, we needed one of these proteins. We needed permease so that lactose could get into the cell. That is true, but even without this permease, a little bit of lactose can diffuse into the cell. It's not a lot, but a little bit can get in there. And when that happens, that does cause lactose to bind here. Okay, hopefully you can see here. And it's going to cause this repressor to change shape. Let's look at it here. So lactose present in the cell, okay? If lactose is in the cell, then what's going to happen? The repressor is synthesized because remember, it's always made because it is constitutively expressed because it's just always, always made. But in this example, the repressor is made, but it doesn't bind to the operator because lactose binds to the repressor. And can you see how these little guys were open here, but here they're closed? So what's happened is the binding of lactose has changed the shape of this protein. This protein is no longer able to bind to this operator. This means that RNA polymerase can move through and that we can have transcription of the genes that are involved in lactose metabolism. Okay, I paused for just a second there because I wanted to bring up for you guys an animation to sort of show you what we have just been talking about. This is the animation from your textbook. And the beginning of this animation, the first 52 seconds, focuses on the gentlemen who were involved in discovering and understanding this operon. And we're going to move through on, and this is going to be an explanation of what we just spoke about, which is how the operon actually functions.
An operon is a cluster of bacterial genes along with an adjacent promoter that controls the transcription of those genes. When the genes in an operon are transcribed, a single mRNA is produced for all the genes in that operon. The promoter contains short, specific nucleotide sequences that signal the start of a gene and is the site where RNA polymerase starts transcription. Operators are regions of DNA that interact with regulatory proteins that control the transcription operons. The regulatory gene of the LAC operon produces an mRNA that produces a repressor protein, which can bind to the operator of the LAC operon. The general term for the product of a regulatory gene is a regulatory protein. The LAC regulatory protein is called a repressor because it keeps RNA polymerase from transcribing the structural genes. Thus, the repressor inhibits transcription of the LAC operon. When lactose is present, it binds to the repressor and changes its shape. As a result of this change, the repressor can no longer bind to the operator region. RNA polymerase can then bind to the promoter and transcribe the LAC genes. When the enzymes encoded by the LAC operon are produced, they break down lactose, eventually releasing the repressor to stop additional synthesis of LAC mRNA. Messenger RNA also breaks down after a relatively short amount of time.
Okay, so hopefully that animation kind of brought everything together for you. But before we move ahead, I want to go back through and make sure that we all understand some of the vocabulary that I used because that was a lot. So an operon is a group of genes with a similar function, right? And this is in bacteria. We don't really have operons in eukaryotes. A repressor was a protein that blocked transcription by binding to DNA. The operator is the region of the DNA or the site in the DNA where the repressor binds. And I'm going to put REP for repressor. binds. Now, one definition or one vocabulary term that I didn't use, but I bet you can figure out, is this term inducer. In our example, the inducer molecule was lactose, but in general, what an inducer does is it binds to the repressor to release it from the DNA. And let's see if there's anything else I wanted to mention. And then the last thing I will say is that remember that constitutively expressed constitutive expression means that these are genes that are always active. In other words, these are always being expressed and always result in protein products. Okay. Now, what I just described for you guys with this example, where we have the repressor here, okay, and the repressor is blocking transcription, this is considered negative control of transcription, okay? And it's considered negative control because it is turning, in its normal sense, it is turning transcription off. Okay. Yes, we can turn it on with the action of an inducer. Remember, lactose functions as our inducer molecule here, but in general, this is negative control. So now what I'd like to do is I'd like to switch gears and I'd like to think about this issue of positive control. And to describe that, I want to go back to our little Petri dish experiment that I mentioned earlier. So earlier I told you that researchers found that if they grew bacteria in a medium that only contained glucose available as a food source, what they found was that those bacteria had no permease and no beta-gal. So I'm gonna abbreviate that, no beta-galactosidase. If they grew these bacteria in a medium or media that had lactose only, then they found that they did have permease and they also did have beta-galactosidase. What's interesting is when you grow these bacteria in a media that contains both glucose and lactose, if these bacteria have both glucose and lactose available, they will not make permease and they will not make beta-galactosidase. In other words, they don't make either of these protein products that help to metabolize lactose. And what that means is that bacteria will preferentially use glucose. Even if there is other food available, bacteria will use the glucose up. And that's because, as I said earlier, that feeds straight into this process of glycolysis, where the bacteria can break down the glucose to turn it into ATP, okay? So let's focus on this situation a little bit here to kind of understand how we might have some positive control of our transcription. Okay, so let me just really quickly preview where we're heading here. Right here. Okay. So what's going to happen? Let's go back and let's draw our DNA as we did before. So here's our DNA. Here's a promoter. Here is the lac-I. Over here is going to be our second promoter. This is going to be our operator. And then this is going to be the 3 genes we're interested in: LAC-Z, Y, and LAC-A. Okay, and in this situation, what I want to do is I want to show you what happens if we have glucose plus lactose available to the cell. Okay, so first thing that's going to happen is we are going to have transcription. I'm just going to draw everything in one color real quick for quickness. So here's our sigma. It's bound to our promoter. And then along with that is our RNA polymerase. RNA polymerase moves through here to transcribe this gene, and ultimately we end up with a repressor protein. And let me color that in really quickly. Oops, here we go, draw. I'm gonna just color this guy in really fast. There's our repressor. And you guys know that usually what would happen is that this repressor would come over here and bind to this operator, right? Normally that's what would happen. But in this example, we have lactose present in the cell. So lactose is going to bind to this repressor. And so instead of having an opening here where it can bind to the DNA, this lactose is going to actually change the shape, I'm going to draw it like this, of our repressor so that it can't bind to the DNA. Let me make this fat so I can color it in. Make it fatter. Here we go. This will color in really nicely. So hopefully you can see that what's happened here is that our lactose has bound. It's changed the shape of the repressor, and consequently, this repressor no longer can bind to the operator. But here's what's interesting. So what that means is that we can now have, let me change the size here, but we can now have, let's see, sigma combined here, RNA polymerase combined there, and we can have transcription of our genes occur. But what we found through experimental evidence is that even though we know that this guy in this situation is not binding to the DNA, and so transcription should be occurring, we still found that when glucose was present, when we had these two together, even though we have lactose and we have the ability to transcribe, we still do not get either of these gene products. We don't get permease, and we don't get, beta-galactosidase. And I'm abbreviating that beta-gal. Okay, so we're still not getting it. Why is that? Well, it turns out that what happens in this situation is our transcription is very slow. So this transcription is happening, but it's happening so slowly that we don't really see an accumulation of permease and beta-galactosidase proteins, because this is just not happening very rapidly at all. To speed up the process, we actually need something called an activator, okay? And in this example, our activator is something called CAP. CAP stands for, I'm going to put this here, CAP stands for catabolite, activator, protein. So CAP. So Here is what happens with this activator, this CAP molecule. It actually has a binding site right upstream of the promoter, so you can see that right here. And it turns out that when glucose is high outside the cell, cyclic AMP is low inside the cell, okay? But when glucose is low outside the cell, which means that it's not there, okay, cyclic AMP is high inside the cell. So in this example, we have a lot of glucose present here, right, initially, and a lot of lactose present. So that would correspond to this situation number one. And what's happening in situation number one is because we have a lot of glucose available, we have very little cyclic AMP available. Now, don't worry about why this is, but y'all remember cyclic AMP. It was a second messenger molecule when we studied cell signaling, okay? So it's used for a lot of different things in the cell. It is a variation, it's like ATP, only it's AMP, and it does have a ring structure attached to it. Now, cyclic AMP, as I mentioned, does a lot of things in the cell. One of the things that it does is it can bind to this protein. When there's not very much of this cyclic AMP in the cell, we have no cyclic AMP bound here. This protein has a shape and where it's not capable of binding to the DNA. But now when glucose goes down, when there's no glucose present, what happens is the cyclic AMP binds here and here. That changes the shape of this protein. Now this cyclic AMP bound CAP molecule is able to grab onto the DNA, and it actually enhances the activity of RNA polymerase to speed up transcription. So let's go back to my previous drawing. In this situation, we had a lot of glucose present, and what we had is lactose as well. Lactose made sure there was nothing blocking RNA polymerase. RNA polymerase could move through here, but it didn't move through very well. It was very, very slow. But if we get rid of this glucose, okay, we get rid of this glucose, what that does is it increases the concentration of cyclic AMP in the cell. Cyclic AMP binds to this CAP protein. When that happens, the CAP protein binds to the DNA. So I'm just going to draw it as a little square here. And CAP protein, sort of, I like to think of it as that it functions like a gas pedal in a car. It works to speed up, it kind of pushes RNA polymerase to speed up this transcription here. And in doing so, we end up with lots of permease and lots of beta-galactosidase. So this situation explains what was happening experimentally. In this example, we had a repressor blocking transcription here when we only had glucose. Here we have no repressor, but that didn't help us out because we also had no CAP protein bound. Okay, so we still ended up with the situation here. But in situation 3, where we have lactose only, we have no repressor blocking transcription, and we have our CAP protein bound so we can speed up transcription, okay? So because this system is turning transcription on, or turning transcription up might be a better way to think of it, we consider this positive control of transcription. Positive, speeding up. Oops, speeding up transcription. Okay, so this system of the lac operon has been really important in helping biologists understand a little bit about gene expression. And remember, gene expression is all about when and how much active protein product is made. Okay, so this system helped us to understand a little bit about when and how much active proteins were made in order to metabolize lactose. And even though it was just one example, it provided us with really 4 big insights into how gene expression works. So let's think about what these 4 key insights are. The first is that many bacterial genes are controlled by operons. And I'm going to show you one last example just to prove my point. I'm sorry, not by operons. What I want to say here is by negative repression of transcription, okay? The second thing that we learned is that we can have activators that speed up transcription. Okay. By enhancing the binding of RNA polymerase. They basically allow RNA polymerase to bind better than it did before. Okay. The third thing that we learned is that gene transcription is regulated by physical contact between, I'm going to put BW, regulatory proteins and DNA. And then finally, the last thing that I want to mention is that post-transcriptional control, allows rapid responses. Now, this is one I forgot to mention, and I want to go back to in our diagram. Let's go back here and think about this repressor. This repressor here has already been translated and transcribed, okay? It has already been transcribed and translated. So this has already been manufactured, it's already been translated, but we have exerted post-translational control upon it by modifying it via binding of this lactose molecule. So we go and spend the energy of making this protein all the time. But in doing that, having this protein sitting around, we're able to modify this protein on the fly, so to speak, when lactose is present, to change its conformation and allow the cell to rapidly respond to new conditions, okay? The same would be true here of our cap protein, okay? Our cap protein, we make it all the time. I say we, I mean bacteria, make it all the time. Here's what it ordinarily looks like. But by modifying this protein using this cyclic AMP molecule to modify this protein. We change its function, okay? So this is an example of post-translational control over gene products. These proteins are gene products, but we go through all of the work of transcribing and translating them, and then we modify them as needed to have them do work in the cell. So this is a post-translational control. All right, one last thing that I wanted to mention is that the lac operon is not the only set of genes in bacteria that works this way. We see other operons that work very similarly, kind of variations on a theme. So this mechanism for controlling gene expression in prokaryotes is fairly common. A second example of this kind of control of gene expression exists in the TRP operon. TRP is short for the manufacture of an amino acid called tryptophan. You may have heard of this amino acid before. It's present in high amounts in turkey, and it's actually what makes you sleepy after Thanksgiving dinner. Okay? But amino acids are needed by all organisms, including bacteria. And so bacteria make tryptophan, as do lots and lots of other critters. So let's look at what happens when we're transcribing genes to make tryptophan, okay? So we're gonna look at genes involved in tryptophan synthesis. So here we have a stretch of DNA, okay, going this way. And on the stretch of DNA, we have a repressor that is bound to an operator, okay? And this repressor is blocking the way for transcription of genes that are related to tryptophan synthesis. That's a P, tryptophan synthesis. Now I want you to notice something about this repressor that's a little different. Tryptophan is already bound to this repressor. So in this example, tryptophan binding to the repressor is actually what makes the repressor the proper shape for binding to the DNA, okay? So when there's lots and lots of tryptophan in the cell, it makes sense that tryptophan binds to this repressor and it stops transcription of genes that would be necessary for making more tryptophan. This tryptophan being here is a signal that there is adequate tryptophan present in the cell and you don't need to make any more. Don't waste your energy. But as the tryptophan in the cell, as those levels begin to drop, what's going to happen is that this tryptophan is going to come off as it gets used for some other cellular process. When this tryptophan comes off, it is a signal that the tryptophan levels in the cell have dropped. And removal of this guy here causes a conformational change here that causes the repressor to no longer bind to the activator. Once that happens, RNA polymerase can move through and transcribe the genes necessary to make more tryptophan. So you can see that this is an example of an operator or an operon. Okay, this is an example of what we call negative control because of transcription, because this repressor is turning off transcription. It's also an example of a negative feedback mechanism. And here's why. Because what's going to happen is as the cell makes more and more tryptophan, so I'm just going to make these little dots be my tryptophan molecules. As the cell makes more and more tryptophan, this tryptophan product is going to bind back to the repressor, which is going to allow the repressor to once again shut down this process. So the products of this process are going to shut down the synthesis of those products, okay? So it is an example of negative feedback. One more thing that it's an example of, remember how I said that adding things to these repressors is an example of post-translational control? Because these repressors have already been translated into protein, but we're busy turning them on and off, making them active, I can't talk and write at the same time, post-translational control. We are busy turning them on and off, making them active and inactive gene products, right? But it's also an example of something that we talked about earlier in the semester called allosteric regulation. Allosteric regulation we talked about whenever we talked about enzymes, but this term allosteric, allo means other, steric means shape. So by binding things to proteins, we can change their shape and thereby regulate their function. Okay, this is everything from chapter 18. Not terribly difficult stuff. Just make sure that you understand these processes. Make sure that you understand how lactose can induce transcription of genes. Make sure that you understand how CAP speeds it up. And then make sure you understand a little bit about how the tryptophan operon works as well. I'll be posting your chapter 19 videos soon. Please let me know if you have any questions, and I'll talk to you soon. Bye-bye.